You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is February 12, 2024. Our topic is Abbas Chapter 7, Immune Receptors and Signal Transduction, Part 2, presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine and Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
Here. So we talked about the T cell receptor and talked about the signaling after that. So similarly for B cell signaling, we want to know about the B cell receptor, right? The structure of it. We know that it's a immunoglobulin that's inserted into the cell membrane of the B cell. So the membrane immunoglobulin has the chains of the immunoglobulin molecule, and then because the cytoplasmic tail is pretty small, it needs the signaling molecules called Ig-beta and Ig-alpha associated with it. Those signaling molecules at their cytoplasmic tail have ITAMs, and those ITAMs are the activating motif, right? The name suggests that immunoreceptor tyrosine based activation motif, and those are important for signaling. So the B cell receptor is associated with the B cell receptor co-receptor, BCR co-receptor, and the co-receptor for T cell is CD4, CD8. Similarly, for BCR, the co-receptor is this whole complex CR2. CR2 also is complement receptor 2, also has a CD marker, CD21, so same thing, CD21, CD19, and CD81. All three of them together are the co-receptor. Now imagine a microbe in the blood circulation, say a polysaccharide, say a pneumococcal bacteria, right? Now that bacteria in the blood is going to be tagged by complement. Complement will get activated and it tags that microbe with its in fragment called C3D. That's a complement fragment that just tags that bacteria or the microbe in question, and that comes across that tagged microbe or antigen is going to be the one that's going to be detected by B cell receptor. So this B cell receptor will bind to one of the epitopes of that whole microbe, right, of that antigen on the microbe. Also, it will, with that C3D bound to it, it can attach to the CD21 or complement receptor 2, that C3D will get attached to that co-receptor. So the antigen is bound to the BCR and the co-receptor using different epitopes, right, or different parts. And then once that co-receptor is activated, it gets the co-receptor activated as well with that C3D component, and at the cytoplasmic tail of the co-receptor is a kinase, non-receptor kinase called LIN. So it is the counterpart of LIC for T cells. So LIC, ZAP70, LAT was the sequence for T cells. Similarly, for B cells, LIN is the one that's going to phosphorylate the ITAMs on the signaling molecules, Ig-alpha, Ig-beta, and that will lead to, and the LIN, there are similar enzymes like LIN called FIN or BLK. So LIN, FIN, or BLK, they will phosphorylate the ITAMs on Ig-alpha, Ig-beta. So that's the first step for B cell activation. All right, so when there is again, we are going through the same process here, multivalent antigen that's gonna help lots of BCRs get connected. There is cross-linking, the co-receptor gets activated, LIN at the cytoplasmic tail of co-receptor will phosphorylate CD19, Ig-alpha, Ig-beta, ITAMs, and also PI3 kinase. LIN once it phosphorylates the items on this Ig alpha, Ig beta, it becomes a docking station for an adapter protein called SYK. SYK phosphorylates becomes a docking station for something called SLP65 or BLINK. BLINK stands for B cell linker protein. It has two names, SLP65 or BLINK. And that is like LAT, right? LAT was attached to the membrane, SLP65 is in the cytoplasm. So a little different, but that's the counterpart of LAT in T cells. So now let's look at the sequence again. Lin phosphorylates the items on Ig alpha, Ig beta. That recruits SYK to its surface, and then SYK becomes it helps with the next step to recruit SLP65. Lin, SYK, blink, and then once that happens, once that SLP65 is connected to that cascade, BTK comes and attaches to it or gets activated at that point as well. So similar to BTK is the ITK in T cells. At this point, the next steps or the cascades move on in 4 different directions that we already talked about. So SLP65 becomes a docking station for GRAB2 SOS that helps with RAS, making RAS GTP and RAC GTP, and that helps in getting ERK and JUNK. So FOS and JUN get created to help with activation of AP1. The second pathway is PLC gamma. PLC gamma activation leads to IP3 and DAG. DAG leads to the activation of PKC beta here, not theta. Theta was T cells, PKC beta is in B cells. That leads to the NF-kappa B pathway. The other pathway, IP3, is the calcium dependent. So IP3 pathway leads to help to open up the intracellular stores of calcium from endoplasmic reticulum, and that pathway is NFAT. So all 3 pathways, along with the PI3 kinase that also gets activated, that helps in survival of the B cells. All right, questions about that? I'm not going to repeat that. We just talked about all those common pathways. Now we move on to the attenuation of immune receptor signaling. So we were talking about activation. How do we make sure there is regulation of the T cell and B cell signaling? So how is it regulated? So there are some inhibitory receptors that are important. We know about CTLA4, PD1. There are also some others like CD94 or NKG2A, KIRS, killer immunoglobulin receptor family. FcR2b is also an inhibitory receptor, and CD22 or Siglec2. All of these are inhibitory receptors. So how do they function? Instead of having items at their cytoplasmic tail, they have items, which means they are inhibitory. They have that inhibitory motif. What happens after that? When the ligand binds to the receptor, there is a similar kinase, right? Cert family kinase like LIC or LIN, and that will phosphorylate the item. So when the item is phosphorylated, it will help in activation of enzymes that are phosphatases instead of kinases. That will lead to dephosphorylation of several different molecules that are in the cascade, and that will lead to dephosphorylated molecules that will help in inhibition of that immune receptor signaling. So items get phosphorylated and then help in activation of phosphatases. Also, that's when these receptors come into play where they can help regulate the signaling. But what happens to the signaling that has already started? There is activation of TCR, there's already ZAP70 that's coming into play, CD3 that's phosphorylated, and all of these phosphorylated molecules will continue to signal in that cell. But there are these molecules called E3 ubiquitin ligases Ubiquitin means it's like a enzyme that uh, helps in tagging the molecules for degradation. And one such in T cells is called CBLB, that tags these phosphorylated molecules like ZAP70, CD3. And so when the CBLB is tags these molecules, the ubiquitin will come and attach to some of these. So it will tag these molecules so that the ZAP70 TCR complex, all of them will be tagged for degradation by the lysosome. All right, moving along. We talked about T cell signaling, we talked about B cell signaling. Now we talk about cytokine receptors. So third part of the chapter. Cytokine receptors, there are lots of cytokines and lots of types of cytokine receptors that bind those cytokines. So our focus is not the cytokines here, we are talking about the receptors that they bind to. So there are families of receptors. There are type 1 cytokine receptor families, type 2. There are other families like TNF receptor family, and we want to kind of know about what is the common feature in some of these families. So cytokine receptor families type 1 have use these signaling molecules called JAK-STAT. There are 3 different type 1 cytokine families. Those are common gamma chain, GM-CSF or common beta chain, and GP130 family. So this is all type 1 cytokine receptors. Then you come to type 2 cytokine receptors that also use JAK-STAT in their signaling. These are the ones that bind to cytokines like interferon alpha, beta, so type 1 interferons, type interferon gamma, IL-10, 20, 22. TNF receptor family binds to these different pathways or are important in the pathways like cascades 8, NF-kappa B, lots of different ligands that they interact with. Then there are families IL-1, IL-17, and then the GPCR family of receptors. I think the most important ones that we want to talk about today are the type 1. So this B part of this figure here that uses JAK-STAT pathway, signaling pathway. So the first one is called the common gamma chain pathway. This can bind to several cytokines. They are numbered interleukin 2, 4, 7, 9, 15, 21. Board question again. So really important to remember 2, 4, 7, 9, 15, and 21. These have one common gamma chain, gamma C, right? Gamma C stands for common here. So common gamma chain, and then the other structure can be different. They can bind to these cytokines. Again, you have to memorize these numbers 2, 4, 7, 9, 15, 21. Common beta chain, GM-CSF, and IL-5. Sorry, go ahead.
I was saying, Raje, that one is a board question, right? Like the common gamma chain?
Yeah.
Yep. Okay.
Yeah. So defect in common gamma chain leads to SCID. The most common form of SCID, so which is X-linked. So again, clinically very relevant. And then GP130 is the third one, where IL-6, IL-11, IL-27 are the cytokines that it will use these kinds of receptors. All right, moving on. We want to know about we'll come back to type 1 and type 2 signaling. That signaling uses JAK-STAT pathway. The third type is the TNF receptor signaling. As the name suggests, this is TNF receptor family. It's a big family. Remember, we talked about another type, one of these families, where we talked about IG superfamily, which means it has the structure has immunoglobulin domain in those molecules, and used by other than immunoglobulin molecules, but they are part of the immunoglobulin superfamily. Similarly, there is this superfamily of TNF receptors, right? They have this particular structure, which is a triad. TNF receptor 1, 2, CD40, FAS, lymphotoxin receptor, BAFF receptors. All of these have this particular structure where there is a triad of trimers, which are like three chains that are bound to each other, and they bind to the ligands that are also supposed to be trimers that can bind to these trimers of the receptor. Once that ligand binds to the receptor, the receptor at the cytoplasmic tail have something called death domain. So that domain then binds to something called like a adapter protein called TRAD, TRADD, TRAD. And that TRAD then binds to TNF receptor associated factors, TRAFs. Those TRAFs then have E3 ligase activity, and then that helps in activation of caspases. Those caspases can lead to apoptosis. So again, death domain, instead of items that we talked about in T and B cell receptor signaling, here we have these death domains that then bind to adapter proteins like TRAD, and then bind to, instead of kinases or other kind of enzymes, here these bind to TRAFs. And then those TRAFs then can activate, again, three different cascades. One of them is caspases that lead to apoptosis. TNF receptors are important, where it says death domain, it leads to that apoptosis, and that's how they use that pathway. But also, apart from them, there are two other pathways that get activated, like AP1 and NF-kappa B. We already talked about those kind of cascades in previously. So, and we talked about AP1 and we'll talk about NF-kappa B in a minute here. And then we know how that helps in the transcription of different genes. So that's how the TNF receptor family works. Now let's think about the type 1 and type 2 cytokine families, right? There, the receptors bind to the cytokine. Say IL-2, right? 247, 9, 15, 21. Remember those. Those are for the type 1 common gamma chain family. Sorry?
If not, you will change other car.
I'm sorry? So when the cytokine binds to these receptors, at the cytoplasmic tail of them, they have Janus kinase, or JAK. JAK are 4 different types, 1, 2, 3. So JAK1, JAK2, JAK3, and the 4th one is called TYK2. Go figure. I don't know how they are named, but those are the 4 JAKs. And those JAK can help in phosphorylating or recruiting STATs. STATs are in the cytoplasm, they are inactivated. When the cytokine binds to these receptors, these can recruit STATs to that cytoplasmic tail, and the JAK will, remember, Janus kinase means it's a kinase. Kinase means it can add phosphate molecule. When they phosphorylate these STAT molecules, these two, so remember they are dimers, right? 2 JAKs are attached to the cytoplasmic tails. They recruit 2 STATs that get phosphorylated, and the 2 phosphorylated STAT molecules can dimerize together. So the 2 STATs come together, and that whole complex of 2 STAT molecules can translocate to the nucleus, where they are going to help with transcription of the genes. So say IL-2 gets attached, there is transcription of the genes that will help in T cell proliferation and survival. What does STAT stand for? Signal transducer and activator of transcription. All right, so for this pathway, the regulation is provided by a few different proteins or molecules called SOCs, suppressors of cytokine signaling, SHP1, SHP2, and then protein inhibitors of STAT. All of these are the inhibitors. All right, moving on to the NF-kappa B pathway. NF-kappa B pathway is common to a lot of different ligands. It could be TLRs, it could be TCR, it could be BCR, it could be a TNF receptor, family receptor. Any of these can lead to the activation of NF-kappa B pathway. The NF-kappa B pathway is 2 different types, canonical pathway and non-canonical pathway. NF-kappa B itself is 2 different types, NF-kappa B1, NF-kappa B2. There are these proteins in NF-kappa B called REL-A and P50. And here in the non-canonical pathway, there is REL-B and P100. So if you read the book, there are more details to it, but I'm going to stick to this figure here for just simplification of explaining what these molecules are. In general, there are different several different molecules, but here are NF-kappa B1, NF-kappa B2. They have these molecules REL-A P50 or REL-B P100. That itself is the NF-kappa B. However, they are bound to its inhibitor. The inhibitor is known as inhibitor of kappa B. So I-kappa B alpha is the inhibitor that's bound. It keeps that NF-kappa B in its inactivated state. Now I'm going to follow the canonical pathway on the left here. So let's look at that. The inhibitor has to be, the NF-kappa B has to be released from its inhibitor so that this transcription factor NF-kappa B can go to the nucleus. For it to go to the nucleus, it has to be released from its inhibitor, and that inhibitor has to be phosphorylated for it to be released from for it to release the NF-kappa B. The enzyme that helps to remove or release the inhibitor of kappa B is called IKK or I-kappa B kinase complex. Again, it's a kinase, so it's going to add a phosphate molecule. So this IKK complex itself has three different parts: IKK alpha, IKK beta, and IKK gamma. IKK gamma part is the regulatory part. It does not really add the phosphate molecule, but it's needed for the function of alpha and beta parts. That IKK gamma is an important molecule also known as NEMO. Defect in NEMO leads to an immunodeficiency called NEMO or NEMO defect. And so again, important to remember that. But this whole structure or complex, so I-kappa B kinase complex or IKK, is gonna be important to phosphorylate, to add a phosphate molecule to the inhibitor of kappa B. When that inhibitor of kappa B is phosphorylated, it gets ubiquinated so that it's gonna be degraded. When that happens, the NF-kappa B part is released and can go to the nucleus so that it can help with transcription. Similarly, on the non-canonical side, it's here IKK beta is important for phosphorylation. On the non-canonical side, it's the IKK alpha that helps to phosphorylate or transform the inactive NF-kappa B to activated form of NF-kappa B, and that gets that can then transfer to the nucleus. Questions at this point? All right, so in the same context, we talked about how there are different ligands that can activate this NF-kappa B pathway. One such pathway to remember is called the KARMA1 MALT1 BCL10 complex. It's not mentioned here, so I can actually go back. If you'll go back and look at the picture that you showed, Ishan, that has that pathway. That's an important pathway because it can also lead to the activation of IKK beta and help in release of canonical NF-kappa B pathway. So let me see if I can show you quickly. All right, here's the pathway. So KARMA1, BCL10, MALT1 that lead to the activation of this whole pathway, NF-kappa B, that helps in the release of I-kappa B. All right. So that's your connection to that whole figure there for NF-kappa B. All right, the last one is, I believe, TGF-beta signaling. So TGF-beta, cytokine can bind to the receptor TGF-beta R2, and then that binds to TGF-beta R1. That helps in recruiting something called RSMED, or receptor-associated SMAD, and then that helps, that binds to CoSMAD, and that RSMAD and CoSMAD together form a complex that become the transcription factor that go to the nucleus and help with the transcription. All right, that's the end of the chapter. Here's a question. Which of the following is an example of GPCR?
E.
E is correct. In the T cell calcium signaling pathway induced by antigen recognition, which enzyme and transcription factor are activated?
E.
E again. All right. I think that's